Good morning, students. Welcome back to Biology class, Chapter 12 Biotechnology and its applications. Biotechnology is the technique of using live organisms or enzymes for products and process useful to humans. Biotechnology deals with the microbe mediated process, in vitro fertilization, synthesis and using of a gene, preparation of DNA vaccines, correcting defecting gene, etc. Biotechnology deals with essentially the industrial scale production of biopharmaceuticals and biologicals using genetically modified microbes, fungi, plants and animals. The applications of biotechnology include therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. Three critical research areas of biotechnology are: first point, providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme. Second point, creating optimal conditions through engineering for a catalyst to act. Third point, downstream processing technologies to purify the protein or the organic compound. Let us learn how human beings have used biotechnology to improve the quality of human life, especially in the field of food production and health. Biotechnological applications. First, we'll see in the agriculture. Let us take a look at the three options that can be thought for increasing food production. First one, agrochemical based agriculture. Second one, organic agriculture. And the third point, genetically engineered crop based agriculture. The Green Revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Increased yield have partially been due to the use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemicals, fertilizers and pesticides. However, for farmers in the developing world, agrochemicals are often too expensive, and further increases in yield with the existing varieties are not possible using conventional breeding. Is there any alternative path that our understanding of genetics can show so that farmers may obtain maximum yield from their field? Is there a way to minimize the use of fertilizers and chemicals so that their harmful effects on the environment are reduced? Use of genetically modified crops is a possible solution. Plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called genetically modified organisms. Genetically modified plants have been useful in many ways. Genetic modification has, the first point, made the crops more tolerant to abiotic stresses like cold, drought, salt, and heat. Genetically modified plants are reduced reliance on chemical pesticides or pest resistant crop. Third point, genetically modified plants help to reduce post harvest losses. Fourth point, genetically modified plants increased efficiency of mineral usage by plants. This prevents early exhaustion of fertility of soil. Fifth point, enhanced nutritional value of food. Example, vitamin A enriched rice. that is called golden rice. In addition to these uses, the sixth point, genetically modified plants has been used to create tailor-made plants to supply alternative resources to industries in the form of starches, fuels and pharmaceuticals. Next point, the seventh point, production of pest-resistant plants which could decrease the amount of pesticide used. Example, Bt toxin is produced by a bacterium called Bacillus thuringiensis, short form Bt. Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide resistance to insects without the need for insecticides. In effect, created a biopesticide. Examples are Bt cotton, Bt corn, rice, tomato, potato and soya bean. Now we'll discuss about only the Bt cotton. Some strains of Bacillus thuringiensis produced proteins that kill certain insects such as lepidopterans, that is tobacco budworm, arbeiworm, etc. Coelopterians, examples beetles, and dipterians, example flies and mosquitoes. Bacillus thuringiensis forms protein crystals during a particular phase of their growth. These crystals contain a toxic that is called insecticidal protein. Why does this toxin do not kill the bacillus? Actually, the Bt toxin protein exists as inactive protoxins, but once an insect ingests the inactive toxin, it is converted into an active form of toxin due to the alkaline pH of the gut, which solubilize the crystals. The activated toxin binds to the surface of midgut epithelial cells and create pores that cause swelling and lysis and eventually cause death of the insect. Specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into the several crop plants such as cotton. The choice of genes depends upon the crop and the targeted pest, as most Bt toxins are insect group specific. The toxin is coded by a gene named Cry. There are a number of proteins are there or the toxins are there. For example, the proteins encoded by the genes are Cry1AC and Cry2AB control the cotton bollworms. That of the Cry1AB controls the corn borer. Next, we'll see the pest-resistant plants. Several nematodes, parasites, a wide variety of plants and animals, including human beings. A nematode, Melodigene incognita, infects the roots of tobacco plants and causes a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference. RNA interference takes place in all eukaryotic organisms as a method of cellular defenses. This method involves silencing of a specific mRNA due to a complementary double-stranded RNA molecule that binds to and prevents translation of the mRNA. That process is called silencing. The source of this complementary RNA could be from an infection by viruses having RNA genomes or mobile genetic elements, that is called transposons, that replicate via an RNA intermediate. Using agarobacterium vectors, nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. These two RNAs, being complementary to each other, formed a double-stranded that initiated RNA interference and thus silenced the specific mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. Thank you.